Welcome to OmegaMatters. I'm Kristina. This is Bill, and today we're talking to Dr. Norman Salem, a prolific omega-3 researcher whose career is focused primarily on DHA in the brain. Dr. Salem obtained his PhD from the University of Rochester School of Medicine and Neurobiology, and for 30 years, Dr. Salem worked at the National Institutes of Health, or NIH, where he led the Laboratory of Membrane Biochemistry and Biophysics. And after retiring from NIH, he worked at Martek, the first algal DHA company, and then for DSM when they acquired Martek. He was the president of ISFAL, the International Society for the Study of Fatty Acids and Lipids, between 2003 and 2006. And he's published about 140 research articles on DHA, which might be a record. And we understand that since his second and final retirement, he's picked up fishing. So Dr. Salem, thank you for coming on OmegaMatters and joining us today.
You're quite welcome.
How's the fishing going?
Well, I'm still working on that. So boats out of the water for the winter.
That's right.
Resume in the spring.
That's great. So we wanted to really dig into DHA in the brain. We have talked about this with some other people, but we're really excited to get into some of the ideas of what we think the mechanics might be. There's a lot we still don't know, but there's a lot that we have learned. So the first simple but big question that we have is, how does DHA get into the brain in the first place?
Well, there's two basic ways to sort of study this. One is our fatty acid compositional studies coupled with controlled diets. And second is the direct study of fatty acid metabolism using tracers. We've used things like deuterium and carbon-13 stable isotopes labeled fatty acids to study, in many cases, in vivo metabolism. So let's start with the compositional studies. I want to tell you about a study done at the NIH lab by Woods. It was a rat feeding study with varying levels of ALA and LA. And it was compared to a dam-reared control, which had the mother, of course, was feeding milk with DHA. So this is early in development. It's an artificial rearing study where you bottle feed them, and you can completely control the diet using purified fatty acids. And we use sort of like a typical American, perhaps you might call it, ratio of 10:1, 10 parts linoleic acid, the N6, 1 part alpha linolenic acid. I'll just call it ALA or 18,3N3, 10:1. Second diet was 1:1 ratio. And the third diet was 1:12 the other way. So it was 1 part LA, 12 parts alpha linolenic. So what we found was that the brain DHA was not as high as the dam-reared diet until you got all the way to the 1:12. So even at 1:1 ratio, which is a lot of ALA, a lot more than typically is taken in by most humans, you couldn't get enough DHA in the brain. You couldn't reach at the same level as the 1% DHA that was in the dam-reared. Now, when you got to 1:12, the brain DHA reached the same level, but by then you had already antagonized the arachidonic acid, so it had fallen fairly significantly. So even that level was not good for the brain. But in the retina, even the 1:12 ratio did not completely bring back the DHA. It was to the same level as when you have preformed DHA in the diet.
Mm-hmm.
So there was the conclusion was that there was no level of ALA that could support a proper ARA and DHA content of the brain and retina in the absence of preformed DHA. Oh. So rats are converting, it's clear that rats are converting quite a lot of ALA to DHA, but not enough to achieve levels equivalent to that of preformed DHA feeding. So let's talk a little bit, that's a rat study. Let's talk a little bit about humans. I'll just deal with the compositional studies by saying this has been extensively reviewed, the human studies by Brenna in an ISFAL policy statement that was published in PLIFA. So there's a series of studies, some using very high levels of ALA, 20, 30, 50 grams a day even. And basically,
Yeah, can you put that in context? How much is that? How much does a person normally eat of ALA?
like it's usually one gram maybe.
Oh, okay. So huge amounts, right?
Yeah, 10, 20, 50 times the normal intake.
Good.
And up to those in some studies. And there was, I believe none of them showed any significant, in adults at least, none showed any significant increase in DHA. Now there were increases in EPA and DPA, but there were no increases in DHA. So
in the brain? In the blood?
This is in the bloodstream.
Okay.
So presumably there's no general increases in the internal organs or the brain.
Yeah.
I think that's an unreasonable conclusion. So let's talk about the tracer studies. We performed in vivo tracer studies using deuterated ALA in both adults and humans. I think about 100 infants were studied and about 50 adult people. The results were that almost every human individual, whether infant or adult, could convert LA to ARA and ALA to DHA. And this is being looked at in the bloodstream. Obviously, we're not able to get into their brains directly.
Right.
But to measure this, you needed a very sensitive mass spec analysis to detect the metabolism. So our first general conclusion is it occurs, but it's not very substantial in humans. We estimated in adults that the conversion of ALA label to DHA, at least as indicated by bloodstream measures, was less than 0.1% of the label had been converted. Now, with controlled diets, one could observe increased metabolism to the long-chain PUFAs when the diet contained low levels of the end products, or put another way, there was less labeled DHA formation when the diet had more DHA. So this makes sense, right? From a teleological point of view. If you take it in, you don't have to make it.
Right, right. Wow.
So there was an adaptation depending on the PUFA levels. Now we had in our adult experiment, about half women and half men. And are you ready for this? All of the adaptation came from the women. There was no adaptation at all in the men.
Yeah.
We were astounded by that.
So when you say adaptation, you mean a drop in synthesis as the diet preformed intake went up?
Exactly.
Okay.
Exactly.
So they have a more sensitive feedback potentially. Or
I could speculate the reasons why, because women, of course, have to support a fetus and have to be able to supply the DHA, have to be able to adapt their metabolism perhaps better. There's hormonal differences, and some of the hormones do, in fact, influence the desaturases.
So we always say that women do it better than men, and people throw around 5% conversion values. So where does that come from?
There are people throwing around a lot of values that are, I think, ridiculous.
Okay.
And I don't think it's anything like 5%. Well, even in a rat, it's about 3%. Okay. We know that from our whole body studies Yu-Hong Lin did. And over a period of 35 days, I think, we looked at every tissue, the whole carcass. And I don't think a human being does a small fraction of what a rat can do.
Okay. So rats are better at it than we are.
or other primates, for that matter. Even dogs and cats can't do very much metabolism.
Mhm.
I want to also say that we did verify this difference between females and males in rats, where we could completely control the diet. By the way, we did control the diet in that study.
Sure.
We had a beef diet in a metabolic kitchen versus a fish diet. And the fish diet, one which I'm talking about, increase in long chain PUFAs, which decreased drastically the metabolism.
Mhm.
And here again, in rats, we did verify that there was an increased level of long chain omega-3s, and there was more stable isotope metabolism. A final note about those human studies with the tracers is that both smoking and alcohol intake lowered the EFA metabolism in the end products. And we found in even our primate studies that chronic alcohol could deplete the brain and the retina of DHA.
Oh, really? Like chronic alcohol from birth, or as an adult, or how did you guys do it?
Yeah, these are adults. These were adult studies primarily.
Okay.
So just feeding it in the water every day over a period of a couple of years.
Oh, wow.
So
Wow.
that's actually fairly small compared to what humans do. We had alcoholics come into our ward drinking, a fifth of liquor every day, or
Wow.
50 beers, living on pizza and candy bars.
Mhm.
So,
do we know if increased omega-3 intake could help reduce addiction?
there's some suggestion of that from Joe Hiblin's studies, but what's more certain is that it can help to prevent some of the pathology, liver pathology, brain pathology, liver pathology in particular. We have some data that's associated with chronic alcohol abuse.
Wow. Wow.
Of course, it can reverse the PUFA losses. And I think we believe that from our studies that those PUFA losses are important in terms for the organ pathology.
Yeah. Yeah.
Wow.
So,
go ahead. So it's going to go back to ALA. I mean, you've said a lot that's really interesting. Just hearing it makes me think ALA just can't provide the DHA for the brain. But then we look at populations around the world that they're vegan, who presumably eat no preformed DHA. How can they have healthy babies? Do they have healthy babies? If they can't be there to make the.
I first want to call your attention. There's a Sanders study, that very old now, 30 plus years ago, where you looked at vegan in the UK, the vegan babies at 6 months of age, and showed that their red blood cells had already had a significant drop in DHA and maybe arachidonic acid also, but DHA for sure. So my expectation is that they can and will accumulate a complement of brain DHA. Both of the mothers can and the infants can, or the children can, but I believe it will be delayed. It will be slower. So because there is metabolism, and in the absence of long chain, the metabolism is greater. So there's that. Often I would expect in many diets, a little bit of DHA creeps into the diets. If you're a vegetarian that still uses dairy, you get some DHA. And that DHA would be, I would be efficiently utilized, I think, and the brain would get some, and retina would get priority. It would get it. So I think that it would be slowly accreted. And we know, for example, from Crawford and Sinclair's classic studies of 39 species, animal species, where about half were herbivores and half were carnivores, that their brain DHA was about the same, but their bloodstream, their liver content was drastically different. So it sort of tells me that a little bit of conversion, a little bit of DHA that they do get in the diet is sucked into the brain, and the brain retains it very well.
Can the brain make DHA out of ALA? No, not the liver. Can the brain do it?
Yeah, it does do it. And we did some cat studies where the cats are said to have no desaturase, delta-6 desaturase. And we found that if you give them a very low EFA diet and you look really carefully with a very sensitive technique, you can see a little bit of ALA conversion to 18,4 and 3, the first desaturation.
First step, okay.
And we noticed that even in these animals, that the stable isotopes were getting incorporated into DHA in the brain. So in almost the complete absence of peripheral metabolism, let's say. Not zero, but it was really in the grasp. So we thought that the brain was, our interpretation of that experiment was that the brain was doing its own metabolism. And we went a little further and suggested that the 22,5N3, which is about the only other omega-3 that you find in the brain in small amounts, very small amounts, might have been the vehicle for transport from the bloodstream into the brain. And then finally, the last step of desaturation occurred in the brain. So that was another pathway that we thought suggested might be operative. Dairy products, beef and things like that have more EPA 22,5N3, and so you can get brain accretion from those sorts of foods.
Okay,
well, that's good. A lot more people eat those.
So, but really coming back, really getting at this question about what quantitatively is the answer for, let's talk about the brain, for the brain, about how much typically under various dietary conditions of the DHA comes from metabolism and how much comes from preformed DHA.
Yeah. Good question.
We thought of a way to quantitatively answer this question. It's in the literature. I like to describe that in a little bit of detail because I think it's important for this discussion. So this was an artificial rearing experiment in rats, bottle feeding, controlled diets, completely controlled fatty acid composition. It was between 8 and 28 days of age. They were given, so we took all of the ALA out of the diet. We gave them pure LA and other non-essential fatty acids. And then we gave all of their ALA as deuterium labeled ALA. And then one group got only that, and the other group got that plus DHA, preformed DHA. So you see then all of the increase in DHA that occurs after day 8 that comes from metabolism is going to contain the deuterium if it comes from ALA, right? And all the increase of unlabeled DHA comes from preformed DHA because the DHA is unlabeled. So we could quantitatively assess during that rapid growth period how much of it came from metabolism or preformed. So the answer was 88% of the brain DHA was derived from the preformed DHA at the end of the experiment 3 weeks later, when it was available and preformed. About 10% then came from ALA metabolism. It was interesting that it surprised us, but just about every other organ in the body, the liver, every other organ, and we looked at 24 of them, I think, tissues, were the same. About within 1 or 2%, 90% of the DHA came from preformed DHA, where DHA was available. 10% from metabolism. Now, what about the animals that did not get any DHA? This was another very surprising thing for me. about 60% of the brain DHA was labeled in that group. 40% still came from non-labeled DHA, even though none was fed.
Really?
This means that it was stealing it from other organs. And in fact, we found that at the end of the experiment, the ALA group, the ALA only group, we looked at the total body and there was no increase, no net increase in DHA in the body. They had the same amount after 3 weeks that they started with at day 8. But the brain, retina, testes, and skeletal muscle and brown adipose all accumulated DHA. And this, and at whose expense? The liver, the digestive tract, bone, skin, and adipose all lost DHA during this rapid growth. The organs were growing rapidly, right?
Yeah.
But they actually had less DHA at the end, even though they were there.
So was that DHA pre-existent at day 8 from their in utero time?
Right. Yeah, they have some from their mother. And
It started off with some low, but there was no net increase over the experimental period. It was just a shift.
Correct.
Wow.
I think this is very scary. Think about ALA infant formula. And we fed that for decades in this United States till the early 2000s. And I believe there are still around the world some formulas without DHA, ARA, DHA that have only ALA. That means that those babies, I would surmise that those babies' organs, as they grow, are losing DHA, and it's being shuttled into these other tissues, which seem to hold on to it better. It's probably they appear to have a higher priority for the DHA that's around. So I thought that was very alarming. So I would say, again, DHA can be slowly accreted from tissues, but it's pretty clear that where DHA is available, 90% of it in the organs comes from preformed.
Yeah, very interesting. Makes me think about my own upbringing. When I was born in 49, and I know my mom didn't breastfeed me. I was just given, this standard formula. Makes me wonder how much smarter I'd be today if she had been a little more careful.
Well, your brain, I think, caught up. Whether or not you were a little delayed, whether you could have done a little better.
Could have been better.
And then again, I'd worry more about your liver and some of the other tissues which were depleted in order to supply your brain.
Oh yeah, that's more your point. That's good. That's interesting.
Yeah.
Wow.
That is really interesting.
Well,
I just
kind of we better wrap up. This is cool stuff, but we don't want to get too long. We might have to have Dr. Salem back for some other questions we've got here.
Yeah. This has been pretty fascinating, and just thinking about how the body uses what it has and does the best it can do with what we give it. And we're trying to figure out the best way to feel good and have proper working brains and feed our babies. So, and I know that was your life's work basically. So we thank you for that. Thank you for coming and giving us all these interesting studies to think about, and for putting it into context. So I think that's it for today.
Yeah. Thank you, Norm.
My pleasure.
Thank you so much.
Very good.